Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Inducible operon. If I say that E. coli has an inducible operon, for its Lac Operon segment, it has an inducible operon. In simple language, if you talk about inducible operons, these are the segments, or you can say proteins, which actually start the expression of a gene. Now we all know that genes contain information to code for a particular protein. The process of replication, transcription and translation is correlated, and during the process of transcription, mRNA or messenger RNA is formed. Now this messenger RNA will have genes or genetic information in the triplet of 3, and now these genes will help to form a polypeptide chain. When I say polypeptide chain, they will join the amino acids, and with the help of a amide linkage, and a polypeptide is formed. So does this polypeptide synthesis is only done by genes? Probably not. There are 3 different types of RNA even involved there: the mRNA, rRNA and tRNA. mRNA is the messenger RNA, whereas rRNA is the ribosomal and tRNA is the transfer RNA. And then your protein is synthesized. But for a protein synthesis to go that far, inducible operons are necessary. In case an inducible operon is absent in a segment of a gene or in an operon segment, what would happen? If that occurs, then your RNA polymerase can never bind to the promoter region. And if RNA polymerase cannot bind to the promoter region, then the promoter region will not be forming the mRNA or messenger RNA, and further proteins won't be synthesized. So any delay or destruction or nucleotide change in inducible operons, or the operons which switch on the mechanism of protein synthesis, can hamper this process. Now, Lac Operon or lactose operon has an inducible operon, which we have studied, which help to convert lactose into glucose. The metabolism of lactose utilization in Escherichia coli is regulated by this inducible operon along with a repressor protein also. Inducible operons, or operons that trigger on or start on the process of protein synthesis. Now, we all know that they contain a gene that encodes for an enzyme which is involved in metabolic pathways. Every inducible operon will consist of a segment of a gene, not only one gene, and this segments of gene will control a certain metabolic pathway. It could be an anabolic pathway or a catabolic pathway, depending upon. It could be your circulation, your enzyme forming process, or your hormone forming process, depending upon whether it's a eukaryote or a prokaryote. Now, the expression of genes is controlled by the substrate of the pathway. This is moreover true in prokaryotes. For example, if we talk about the lactose metabolism, if lactose as a substrate is not present in the media or inside the cell, then obviously the expression of genes will have no use. Those proteins or enzymes formed will act on which substrate and what is their function. An example of inducible operon is the Lac Operon, or you say lactose operon, which encodes the enzymes, or which forms the enzyme required for metabolic processes, especially in Escherichia coli during food utilization or lactose utilization. In some cells, if you talk about, certain enzymes are normally present all the time and they are called as constitutive enzymes or constitutive organelles or certain proteins which are present in certain organelles. While certain enzymes are synthesized only when they are needed inside a body. For example, certain gametes or certain reproductive cells and certain enzymes which are related to zygote formation in human beings will only be synthesized when the process of fertilization will take place. That proteins will only be synthesized when a new progeny would be formed during the process of fertilization. In a young adult or in a teenager, those proteins will never be synthesized. They are synthesized only after a certain age group and especially during the process of fertilization. Now, the synthesis is induced by a special substance which is termed as inducer, or you can say the on button of any regulation mechanism or gene expression mechanism. Now this on button inducer is very specific in nature. For example, lactose in E. coli will have a specific inducer. Your proteins in human body, that's insulin, will have a specific inducer. The mechanism through which inducers work is nothing but genetic induction or the on process mechanism. Students, in this part of the chapter, we did study about inducible operons, or you'd say the operons or machinery which start the process of gene expression. And every individual, whether it is your eukaryotes or whether it is your prokaryotes, will have an inducible mechanism which will work with the repressor mechanism and co-regulate the process of protein synthesis depending upon the situation and the requirement. I hope you all are clear with this concept of inducible operons. Thank you.